With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, have a look at the question. Question is unique restriction sites are present in small region of DNA in a vector. What is this region called? This region is called as a multiple cloning site. Multiple cloning site or MCS, or short form it is called as the MCS. MCS is the multiple cloning site, also called as a polylinker. It is also called as a polylinker. It is a small region of DNA or short segment of DNA. We can say that short segment of DNA which contains very many restriction sites. It contains many restriction sites, and these many could be up to approximately 20 restriction sites are present. Restriction sites, and this is a standard feature of the engineering plasmid. So if you see these restriction sites, these are unique to a particular plasmid. Here we are taking the example of the PBR322 plasmid. Its total size is 4361 base pair. Here if we can see that the various restriction sites present are the PST1, ECO R1 restriction enzyme, HIND3, ECO RV, BAM H1, CEL H1, NDE. So these are the different enzymes, restriction enzymes, whose restriction sites are present in this vector. And these restrictions enzyme also encode for certain. Here we can say that the ampicillin resistant gene, that this site is present in this, so this act as a selectable marker. Similarly, tetracycline, in the tetracycline there are different restriction sites are present, so it means if there will be cut, so it will tetracycline will not react, so definitely it act as a selectable marker. So you can notice here all over there are different positions. Restriction sites are present, but there is no restriction sites is present at the ORI. So it can't be present, because if the ORI contain any restriction site, that will cut the DNA and it will disrupt the major function of the major characteristic which if of the vector is to replicate independently, that will disturb. So ORI always, here we can say that ORI always doesn't gain, ORI that is origin of replication, always can't have any restriction site, rest all selectable markers can have that. Now what is the function of having these restriction sites? So these restriction sites are a useful feature which allow the insertion of foreign DNA without disrupting the rest of plasmid, because it already they are having, so they don't need to disrupt the whole plasmid, need to disrupt the whole plasmid, or we can say that to make special changes, because already it contain different restriction sites. So it just they have to add that enzyme and the cut has to be made, that is act as a selectable marker. That cut will lead to the selectable marker. Suppose there is a cut in the tetracycline and then ampicillin will added that particular clone which is having restriction cut for the tetracycline, they won't act as a tetracycline resistant, so it act as a selectable marker. So in the question they have simply asked unique restriction sites are present in small region of DNA, so we can say that our answer is that is multiple cloning site, or you can also write it as a polylinker. So these are multiple cloning site. Hope you get it. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.